Hello students, Biosavers are back again with an interesting topic in the cell biology series that is about cell wall. So let's get started. So cell wall is the main feature or the organelle that distinguishes plant and animal cell. It is present in all plant cell, most fungi and protists, while animal cell lacks the cell wall. So looking into the plant cell wall, its thickness ranges from zero point 0.1 micrometer to 10 micrometer while it is secreted by the cytoplasm. It is mainly composed of four chemical constituents, that is cellulose, hemicellulose, proteins and pectins. Cellulose, it is an unbranched chain of glucose molecules. There are 6,000 glucose molecules in each chain and many such chains combine and form a bundle-like structure which is called as microfibrils, and these microfibrils are held together by the hydrogen bonds. Second is hemicellulose. It is branched polysaccharide which is having main chain of sugar such as glucose molecules and side chain of other sugars such as xylose molecules. Pectins, it is branched or unbranched polysaccharide which is rich in galactonic acid, and when plant cell is attacked by pathogens, fragments of pectins they trigger the defensive response by plant cell. Proteins, they interweave through the cellulose and pectin networks. Other than that, cell wall is well hydrated and it has 60 to 70% of water content in its mass. Physically, there are three layers of cell wall. Middle lamella, which is the first structure that is formed between newly formed daughter cells at the time of cytokinesis. It is generally made up of calcium pectide and magnesium pectide. Then primary wall. After the middle lamella formation, it is the first formed wall. Growth of primary wall occurs due to incorporation of material within the existing wall. Such mode of growth is called as intussuspension. Then secondary wall. In mature cells, more layers of wall material are added internal to the primary wall. These layers are called as secondary wall. And growth by addition of new wall material on the existing one, it is called as accretion. So as seen here, primary wall is formed over the middle lamella, and it is the middle lamella which make contacts with the another cell, that is middle lamella is exposed to the outer surface. While secondary wall is usually consists of three sublayers, that is S1, S2 and S3. This formation of secondary wall is not uniform in all the cells, which results in the formation of tissues such as parenchyma, sclerenchyma, collenchyma, fibers, vessels and tracheids. Now in gymnosperms, that is tracheids of gymnosperm form another layer of wall material internal to the secondary wall. This layer is called as tertiary cell wall. It is made up of cellulose and xylem. Apart from this, cell wall have various other structures like pits. At certain places, secondary wall is not present. Such areas are called as simple pits. Pits are separated from each other by middle lamella and primary wall. So area or the wall between two pits is called as pit membrane. Along this membrane, materials can pass. While in gymnosperms, the secondary wall partly overhangs, that is covers the pit. Such pits are called as bordered pits. Then there are certain additional materials which may be deposited in the cell wall for specified purposes. These depositions can be of lignin, which is a tough glue-like substance. It is generally impregnated in the primary and the secondary cell wall of woody plants. Then there is cutin, which is a fat-like substance, and it is deposited on the exposed surface of cell called cuticle. This makes the cell wall relatively impermeable to water. Same like cutin, suberin is also a fat-like material and it is also deposited in the secondary wall of cork cells. And it also makes cell wall impermeable to the water. And then there is silica, which is deposited in the cell wall of grasses. Some other substances such as minerals, waxes, tannins, resins, gums, etc. may also be deposited. So now then there are certain places where secondary wall is not formed. So such areas have fine simple or branched canals in primary wall and the middle lamella. This canal acts as intercellular connection, which means it connects the internal content of two cells. So through these canals, plasma membrane, cytoplasm and endoplasmic reticulum of adjacent cells are continuous. It is called as plasmodesmata, and the endoplasmic tubules extending through them are called as desmotubules. Now through this connection, both living and non-living matter are present. Living matter present in this connection is known as symplasm, and the non-living matter present in this connection are called as apoplasm. So now looking into the fungal cell wall, they have same cell wall structure, just the wall is made up of chitin instead of cellulose molecule. Similarly, prokaryotes have peptidoglycan in their cell wall structure instead of cellulose. It may also contain teichoic acid. while physically the structure is same. So now, as we have studied the cell wall structure in detail, we can easily describe its function. That, as it is the outermost structure of cell, it maintains the cell shape, provides support, and also protects it from pathogens. Looking into the chemical structure, we can say that many enzymatic activities are associated with the cell wall, and it also balances the internal osmotic pressure. Then, with the help of physical structure, it prevents undue expansion and exchange of materials. And finally, the other structures such as pits, plasmodesmata, depositions link up the different cells and thus allow system combination. So that's it for today. I hope now you're very clear with the cell wall structure. To check that, you can also go through a small quiz which I have provided in the description. If any queries, do let me know in the comment section. Thank you.